Fossilized ear bones, known as otoliths, are revolutionizing our understanding of freshwater fish evolution. These tiny structures, once hidden in ancient sediments, offer clues about species that lived millions of years ago. By analyzing their shape and composition, scientists can identify long-lost fish and trace their migration patterns. Remarkably, these discoveries challenge previous assumptions, revealing more complex and diverse histories than ever imagined. Otoliths are rewriting the story of freshwater ecosystems, one bone at a time. When saltwater fish long ago evolved to live in freshwater, many of them also evolved a more sophisticated hearing system, including middle ear bones similar to those in humans. 2/3 of all freshwater fish today, including more than 10,000 species, from catfish to popular aquarium fish like tetras and zebrafish, have this middle ear system, called the Weberian apparatus, which allows them to hear sounds at much higher frequencies than most ocean fish can, with a range close to that of humans. University of California, Berkeley paleontologist Juan Liu has now used the structure of this Weberian apparatus in a newly discovered fossil fish to revise the origin story for the evolution of freshwater fish. Fish with a Weberian ear system, referred to as otophysan fish, were thought to have moved into freshwater approximately 180 million years ago, before the supercontinent of Pangaea had broken up into the continents we see today. Based on Liu's new timeline, they now appear to have arisen much later, about 154 million years ago, during the late Jurassic period, after the beginning of Pangaea's breakup, and coinciding with the appearance of today's oceans. Liu's analysis of fossil and genomic data implies that the fish originally developed precursor bones of their superb hearing while still in the ocean. Only later did they develop fully functional enhanced hearing, after the two separate lineages moved into fresh water, one evolving into today's catfish, knifefish, and African and South American tetras, the other evolving into the largest order of freshwater fish, the carp, suckers, minnows, and zebrafish. The marine environment is the cradle of a lot of vertebrates, said Liu, an assistant adjunct professor of integrative biology and an assistant curator in the UC Museum of Paleontology. A long-time consensus was that these bony fish had a single freshwater origin in the large continent Pangaea and then dispersed with the separation of different continents. My team's analysis of some fantastic fossils that shed new light on the evolutionary history of freshwater fish and found completely different results. The most recent common ancestor of otophysan fish was a marine lineage, and there were at least 2 freshwater incursions after that lineage split up. This finding reshapes our understanding of the evolutionary history and intricate biogeography of the world's most successful group of freshwater fish, she added. These repeated incursions into freshwater at the early divergence stage likely accelerated speciation and are key factors in explaining the extraordinary hyperdiversity of otophysans in modern freshwater faunas. Liu and her colleagues describe and name the 67-million-year-old fossil fish, Acronychthys macagnoi, in a paper published in the journal Science. In that paper, the researchers analysed 3D scans of the fossil's Weberian structure and the genomes and morphology of modern fish to revise the genealogy of freshwater fish and also simulate the frequency response of the fossil fish's middle ear structure. A 3D model of the head of the newly named 67-million-year-old fossil fish, Acronychthys macagnoi, based on CT scans. The skull bones are brightly coloured while the ribs and backbones of the spine are in grey. The small, bright red bones at the junction between the spine and head are ossicles of the Weberian apparatus. Credit: Juan Liu, UC Museum of Paleontology and Don Brinkman, Royal Tyrrell Museum.